So it's my pleasure to welcome you all again to the second India Science Festival. TIGS is a sponsor of this event, and I had the pleasure of coming there and talking to a very big audience in Pune in January of 2020. This year, the theme for our panel discussion is selected advances in modern biology. Now, there's so many exciting advances in modern biology. I could have chosen 1,000 vignettes, but our time is limited. And I chose a set of experts here who are passionate about their fields, who made significant contributions. And most importantly, their passion is infectious. So I hope you will get the sense that this is a group that will excite you, stimulate you, and get you interested in science, because that's the whole goal of the India Science Festival. So without further ado, let me just introduce the panelists. We will have 4 panelists. The first one is Dr. Vivek Malhotra. He's a group leader at the Center for Genomic Regulation in Barcelona, Spain. He's one of the world's experts on protein trafficking and cellular compartmentalization, how subcellular organelles are formed and what they do. But today he will talk to us about controlling mucin levels, mucin secretion in the lung airways and colon. And this is very important for cystic fibrosis and diseases like colon cancer. After that, we will have Dr. Sonia Sen, who is a senior scientist at the Tata Institute for Genetics and Society. She's an expert on neurodevelopment and behavior, particularly working with insects. And she's got a very ambitious topic. In 7 minutes, she's going to tell us how to build a brain. And luckily, it's a fly brain, not a human brain, but it'll be exciting all the same. Then we have Dr. Amita Sehgal, who's a John Hermesser professor in the Department of Neuroscience at UPenn. And she is one of the world's experts on circadian rhythm and sleep in fruit flies and mammals. And she will tell us everything you want to know about why organisms need sleep and what happens if your sleep cycle is disrupted and you have things like jet lag or disrupted sleep cycles. And finally, to end the session, we will have Dr. Venkatesan Sundaresan, who's a distinguished professor from the Department of Plant Biology at UC Davis. He is a world expert on plant reproduction, particularly in rice, and he will talk to us about genome editing of crop plants to meet food needs through clonal hybrids. So what I'm going to do is to have these panelists talk in this particular order. Each one of them will talk for about 5 to 7 minutes each. And I've basically asked them to try and address what their fields of biology is, what model organisms do they work with, and why their particular model organism allows them to address that particular problem in biology. But more broadly, they will tell you about their research, how they got interested in this topic, what are the most exciting advances, and what the potential implications are. So it's a broad palette. I'll let each one of them excite you in their own way. So with that, Vivek, I'd like to lead this off and have you start your presentation.
Okay, but it says that I cannot start my video because the host has stopped it.
No, we can.
Can you see my slides?
Yeah, we can see your slide.
And you hear me? Oh, all right, okay.
Yes, yeah,
okay.
yeah, you're
right.
fine.
Okay. So good morning, afternoon, evening, wherever you are. Thank you, Suresh, for this wonderful opportunity. So I've been given 5 to 7 minutes to present a topic that is of great interest to me. And this is something that we've started working on for, I'd say, about 5, 6 years ago. So this is one of the themes of my lab. And it has to do with how cells secrete mucins and how the quantities and the quality of mucins secreted is regulated. So human beings secrete a liter of mucins per day. That's a lot of mucins. And these mucins, when they are secreted by specialized cells that line all the way from your nose, your airway, lungs, intestine, all the way to the very end, these specialized cells called the goblet cells release mucins. These are heavily glycosylated proteins. And when the mucins come out, they mix with liquid and ions in the extracellular space, and it becomes mucus. And this is something that you are probably all familiar with, because this is something that comes out of your nose when you have a terrible cold or when you sneeze. Now, this is in fact a very good thing to have. We secrete mucins, which become mucus, and this provides the first layer of defense from foreign pathogens. So the lining of your airway and the intestine has a very thick layer of mucus, and its function is to basically only allow gases to go across. Everything else is prevented, and the mucin secreting cells, as shown here in pink, they're usually surrounded by ciliary cells. So mucus comes out, and anything that attaches to it, bacteria, viruses, pathogens, etc., 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 the movement of the cilia causes the mucin layer to move, and it is removed from the body in both directions. Okay. Now, everyone knows about Sars virus, and one of the things that the viruses like the SARS do is they prevent mucin secretion, and this is something that we are trying to address, which makes them more infective. Okay, because now they can penetrate this barrier in the airway and in the intestine. Now you need to control the quantity secreted, because if you secrete too much, you end up with problems of the airway, such as chronic obstructive pulmonary diseases, asthma, and cystic fibrosis. And if you don't secrete, then in the intestine, you have problems of pathologies like the irritable bowel disease, Crohn's disease, colitis. And it turns out that there are these mucinous cancers. So for example, one of the issues that we are trying to address is patients with mucinous colorectal cancer. So you have cancer cells that build a thick layer of mucus around them, and those cells become chemo and immune refractory, meaning they are basically now living in hiding. And these are the cells that become usually very, very, very aggressive. And in fact, one of my students, who was a professor at Harvard, died at the age of 41 last year as a result of this mucinous colorectal cancer. So the question is, how does a cell control how much mucin is secreted at any given time? And I'm going to give you an example here of how we can study these processes. So these are specialized goblet cells that are grown on tissue culture plates. And usually there is some mucin secreted at all the time, and it's called the basal stimulation. But when there is an agonist, which in our case, we just simply use ATP, it could be cigarette smoke, it could be a pathogen. What happens is there is a burst of release. And in some cases, you can use very, very artificial conditions, which causes massive release of mucin. So I'm going to try to see if this a movie would work. So on the slide which says no stimulation, the bright particles that you see are many cells that are filled with these mucin granules, okay? Now, if I were to treat these cells, hit these cells with ATP on the left column, which you will see ATP, you see this material coming out, which is kind of light in color. And if you stimulate the cells with PMA or foveal, this is what would happen if there was a pathogen attacking these cells, you will get a massive release, okay? So the question is, how does a cell release just the right amount of mucins? And for the last 4 or 5 years, we've been trying to address this issue by identifying the genes or the braking mechanism which controls this release propensity. And we have identified, and this is just to summarize 5 or 6 years worth of work in one slide. So what we've done is we've used these airway cells and cells of the colon and done a genome-wide screen to identify all the genes that are required for mucin secretion, production and secretion. So what we have found is a protein called KChIP3, which is basically a potassium channel interacting protein number 3, which is recruited to a granule that forms from the Golgi apparatus. So when the proteins are secreted, they're made in the endoplasmic reticulum, they go to the Golgi, where they get glycosylated. From the Golgi, they are packed into specialized granules, which are first given the name immature granules. These granules then mature by a very inside the cell till a signal tells them to go and fuse to the surface and release their content. Well, it turns out that as long as these mature granules contain KChIP3 on the surface, they cannot fuse. But if you remove it, then these granules go on to fuse. So this is basically the brake. This is the traffic light. So you're quite familiar with the traffic signals in India. You come to a traffic light, there is a red signal and you stop. Now, when the signal goes green, everyone rushes through, okay? And this is that signal. K-CHIP3 is that signal that prevents granules from fusing. But the question here is, why do some granules contain K-CHIP3 and why don't the others not? And this is something that we are trying to address. But for the time being, this K-CHIP3 provides a great handle to understand and to address how cells control the amount of mucin secreted. And in the next slide, I just give you an example of the kind of observations that have allowed us to make this statement. So we can do all these experiments in tissue culture cells, but we've also created a mouse knockout in which the K-CHIP3 gene has been deleted. So there is a mouse that does not have K-CHIP3 gene, the mouse is fine, okay? But what we go and then do is to ask, what is the level of mucins? And in this case, we are looking in the colon of the mouse. The wild type normal mouse has this much mucin as shown in this black line here, okay? If you remove this K-CHIP3, then look at the amount of mucin secreted. And we've calculated on average about 2 to 3-fold increase. But this is very, very difficult to quantitate because in some layers, some section, we see about a 2-fold increase, whereas in others, we see 5 to 7-fold increase in the amounts of mucin secreted. Okay. So you can imagine now, if you have this much mucus present around the cells, nothing can penetrate. In fact, in these mice, we often see accumulation or presence of many, many different kinds of pathogens, which we can stain simply by using DNA dyes. We don't know the microbiome of these cells, of these tissue. This is something we are trying to do. But this is, in my opinion, the first understanding of how a protein, a 20 kilodalton, a small protein, can control the propensity of the mucin granules in terms of their release propensity. And so this is something that is ongoing now. And what we have also done, I don't know how much time I have, what we've also done is we've taken cells from patients with colorectal cancer, okay? And we know that these cells are refractory to standard chemotherapeutic agents, okay? What we can do is, ordinarily, if you take a normal cell and if you were to add these chemotherapeutic agents, you kill them. But if we remove K-CHIP3, there is so much mucus secreted, there is a thick layer of mucus around these cells, and when you add those chemotherapeutic agents, they become refractory, meaning they become ineffective. So we are trying to do now the opposite experiment, which is to find ways chemically, and again, I don't have the time for going into the details, we have done a screen of small molecules in collaboration with AstraZeneca, which allows us to inhibit this mucin secretion. And when we inhibit mucin secretion, when we then treat the same cells with chemotherapeutic agents, we can induce rapid killing of the cells. So I think this provides us not only an understanding of how cells control mucin secretion, but we also have the possibility of taking our studies to the next level and at least address issues that have to do with hypermucin secretion related issues. So I think this is all the time I've been allocated, and this would be a right time to stop. And I would gladly answer any questions that you might have. Thank you.
Thank you. And we'll turn this over to Sonia Sen next. Vivek, if you would stop sharing your slides, and Sonia can come on. Yeah, thank you. Sonia, you're on mute.
Okay, can you see my
Yes.
dark screen?
Yes.
Okay. Okay, hello everyone. Hi Suresh, hi to the rest of the panelists and to the folks at the India Science Festival. Thank you so much for the invitation to be at this fantastic forum. I hope that next year we'll be able to see it in person, because I remember from pictures of last year, it's a very vibrant forum. Okay, I will dive straight in and tell you about what I promised. Oops, my slides are not moving. Which is how to build a brain. And this is because our lab basically works on trying to understand how nervous systems are put together during development. And once they're put together, how do they generate behavior? So what do I mean by this phrase, generate behavior? If you think about the animal kingdom around you in your everyday life, we're surrounded by animals that do very interesting things to their environment. So for example, this female mosquito that bit me and I swatted it, or for example, these animals that make these nests to capture prey or to nurse their young. Animals around us really do very interesting things, which is what we want to understand. And it's because of nervous systems. The animal kingdom is the only set of organisms in the entire tree of life that has a nervous system, and therefore has the ability to respond to stimuli in the environment in very exquisite ways, such as these pictures that I'm showing you over here. So that's what we're interested in. How is this nervous system put together during development, and how then does it generate all of these behaviors? So in the lab, we actually use the fruit fly, Drosophila melanogaster, to be able to do this. And there are many reasons why we use the fly. And I'm going to spend a couple of minutes on why that is. So the first reason is that it's just simple to grow. You can basically take solidified porridge and put a few flies there, and they'd be happy to grow in it. So they're quite cheap. And then, people have been studying flies for more than 100 years now. And so that means that we have accumulated this wealth of knowledge and resources that we can use to ask the right questions. And this may surprise some of you, but a lot of the genes that flies have are shared with human beings. The repertoire of genes is the same, although humans may have many more copies of those very same genes. So that's very important. If you want to understand the genetics of any even human biology, flies are a great place to start. And then, of course, because of this history of research with flies, we actually have developed a fantastic resource, an arsenal of tools to allow us to do fantastic experiments with flies. So we can turn on and off genes at will. We can do it at different times. So we can do it in the larva. We can turn on and off genes in the adult. We can do it in specific tissues. So we can say, I want to turn on a gene in the brain, or I want to turn off a gene in the gonad. And you can do that with flies. And so that's really important for the questions related to development. But if you're interested in the nervous system from the perspective of behavior, you can do something else in flies with exquisite detail. You can turn on and off neurons at will. So you can go into the brain, turn on a set of genes, and then ask, what does it do to the behavior that I'm studying? Or you can turn off those neurons and ask the same questions. So really, a wealth of tools that you can use to design the perfect experiment to answer the question that you're interested in. And of course, all of this would not be much use if the fly didn't offer the biology related to the question that you're interested in. So we're talking about the nervous system. And so this is a question that I get probably asked the most amongst family and friends. Do flies have brains? And I want to tell you, yes, they do. They have very complex brains. So they're complex. The brains are complex in terms of numbers of neurons. So there are about 150,000 neurons in the fly. It's complex in terms of its organization, in terms of the connections that they make with each other. And as a result of this complex brain, flies exhibit wonderful behaviors. And you'll hear some of this in Amita's talk next. Flies have the ability to show sleep, circadian biology. Flies show exquisite courtship behavior, as you can see in the stopgap motion picture here. They show aggression, they fly, they can learn, they can form memories. So really, the nervous system is complex enough, yet not too complex, and it has the right set of behaviors to be able to ask interesting questions in. Okay, so let's just look at what we've learned about the brain from the fly. So here's a schematic of what the brain looks like. So that's the purple structure over there in the adult fly. You can see the adult fly in the gray over there. And that nervous system is made up, that's where all your 150,000 neurons are. And those neurons are made from neural stem cells. So here's a picture of 2 neural stem cells sitting in the brain of the fly, of the developing fly, and you can actually see this is a real image. So here's the neural stem cell, and it's budding off a neuron. You can see it in action over here, and here's a video to show you the same. I'm sorry, I seem to have lost the reference there. This is not from our lab, but you can see this is during development. The neural stem cell is dividing and budding off these neurons, so that at the end of larval life of the fly, you get many neural stem cells with clusters of neurons that they have generated sitting around them. And the brain is full, the developing brain is full of these neural stem cells. And now if you allow the fly to go through metamorphosis, that is through the pupil life, then all of these neurons send out processes and find the right partners to make the right connections with. So now if you looked at these in the brain of the adult fly, some of these stem cells would have generated a bunch of neurons that looked like this. Other stem cells would have generated another set of neurons that looked like this. And a third stem cell would have made this set of neurons. So each stem cell is making a different set of neurons. And this is what we've learned from the fly, that you have these modules of development. Stem cells seem to be setting their identity apart. They become different from one another and therefore generating different neurons. And so this is what we're interested in my group. We want to understand how is one stem cell made different from another so that they can generate different neuron types. And you can see how this would be important, because if you understood how a particular class of neurons were made, you would be able to make that neuron type at will. And I'm sure you're joining the dots here now, because in the context of stem cell therapies, you want to be able to make neurons, the specific neurons that are lost or dead in the scenario of disease or injury. You don't want to make any old neuron. You want to make that dopaminergic neuron that is no more, that is depleted from the brain. And so we want to understand how specific neuron types are made so that we could reverse engineer them at will. So that's what half my lab works on. The other half works on behavior, trying to understand how this brain now gives rise to behavior. And for this, actually, we don't use the fly. Well, not entirely. We still rely on the fly to test our ideas and get inspiration and clues. But we actually study another related insect, the mosquito. So the mosquito belongs to the same family as flies. It's a dipteran. So its structure, in terms of structure of its nervous system, it's very similar. In terms of genes, it's very similar. So we use our knowledge from the fly to understand behavior in the mosquito. And the mosquito has exquisite behaviors, the most important of which for society is its blood feeding behavior. So there are very many species of mosquitoes in the world, and not all drink blood. Most don't, in fact. And the ones that do, it's even in those, the male never drinks blood. It's only the female mosquito that drinks blood. And mostly males and females usually will drink nectar or sap. And sometimes, only sometimes, that only the female switches to taking a blood meal. So that's very interesting. We know that the female does this largely because she needs the blood to make eggs. And so here's the ovary of a female mosquito. You can see this bunch of grape-like structure. Each one of these grapes in there is actually a single developing ovary. And that's what she needs the blood meal for, to make it into a full mature egg. And so we want to understand how those circuits in the brain are driving this very important public health impactful behavior, right? What is the circuitry that makes the female mosquito take that decision of, I don't want nectar anymore, I want a drink of blood. Okay, so and you know how this will be important, right? Because if a female mosquito didn't drink blood, she couldn't carry diseases. Okay, so that's the flavor of what our lab does. And with that, I will end and just show you the gang that does all of this work. You can read their names over there. And I will hand over the stage to, I guess, Amita. And I'm looking forward to any questions and discussions in the later sessions.
Thank you, Sonia. So if you would stop sharing your slides, and our next speaker will be Dr. Amita Sehgal. She's going to talk to us about sleep. Amita, you might still be on mute.
Okay, I just wanted to get my slides lined up. Can you hear me?
Yes.
Okay, so I'm really pleased to have this opportunity. Thank you all for tuning in, and thank you, Suresh, for putting this together. Been enjoying the talks that have come before me. So what we work on is circadian rhythms, and these are 24-hour rhythms of behavior and physiology that are seen in almost all living beings. So I think most people are aware that we sleep at a specific time of day, but sleep and wake are not the only aspects of physiology that are under this 24-hour control. Truth of the matter is that it's hard to find an aspect of physiology that doesn't, on some level, show what we call circadian regulation. So here on this slide are shown the different processes in humans that show differences across a 24-hour day. And so, you'll notice that at some times of day, you're going to have changes in blood pressure, melatonin, body temperature, sleep, of course, down here. And what I'd like to stress is that these are just a few of the processes that in humans have a 24-hour component to them. So glucose is different at different times of day in our blood. Insulin is different at different times of day. Cholesterol is different. Okay, our response to drugs is different. Cortisol is different. So almost everything in our bodies is changing over the course of a day-night cycle. So you might say, okay, fine, so this happens, but why should I care? Let things go up and down as they do. And the reason you should care is because when rhythms are disrupted, this internal pattern we have, and this can occur in different ways. So it could be intrinsic factors. There are people who actually don't have very well-defined rhythms, sleep-wake cycles and other rhythms, because they have mutations in them. The other scenario in which rhythms are disrupted is with aging. So you all may have noticed, if you see older relatives, that they are going to sleep at odd times, perhaps waking up in the middle of the night, perhaps napping more during the day. That is all a sign of these rhythmic patterns breaking down with age. But these rhythms can also break down with extrinsic factors. So if you do shift work, for instance, if you work at night instead of during the day, you'll find that you generally don't feel very well. And it's because your body rhythms are not in synchrony with the environment. Chronic jet lag. So if you travel to a different time zone, which many of you may have done, you'll notice that it takes a while to adjust to the new time zone. And while you're adjusting, again, your sleep patterns are off, your body functions are off, your gastrointestinal functions are off, for instance. And then finally, if you eat at the wrong time of day, you also can have your rhythms disrupted. So if you eat really late at night, again, your health is compromised. And when rhythms are disrupted by any of these scenarios, there is an increased propensity towards neurological and psychiatric disease. So depression or neurodegenerative disorders, dementia. And there's also increased susceptibility to metabolic and cardiovascular disease, diabetes. So metabolic diseases in particular are very, very closely linked to circadian clocks because like I mentioned to you, insulin, sugar, and glucose, everything is cycling in our bodies. So these rhythms that I've mentioned are controlled by clocks that are within us. And when I was a trainee at the time, when I had just finished my PhD and I was looking to do my postdoctoral work, I became interested in this question of how these clocks are made within us. And the model that I chose to work with is the one that was just so beautifully introduced by Sonia, which is the fruit fly Drosophila melanogaster. And I don't have to tell you why flies are a great model to work with, because Sonia has done that already. Very glad she went before me. And so what in this case we used the fly for was to understand how a clock is generated. So I joined the lab of Michael Young, who was at Rockefeller University in New York City. And he was using genetic approaches to understand how you make a clock within a fly, because flies also have rhythms and they have, rest activity, very robust rest activity and other rhythms. Now, one of the reasons we were asked in these presentations to talk about how we were drawn to our field of interest and why we chose the approaches we did. And one of the reasons that I was drawn to this was because I had been interested in the genetic basis of behavior. What is it in our genes that makes us behave in a particular way? And it seemed to me that rest activity behavior, which is a very, very robust, well-defined behavior, might be very tractable. And so it might lend itself better to genetic analysis than would a lot of other behaviors, such as learning and memory, for instance. And so this is how I came to be in this lab. And we took genetic approaches to find genes that generate a circadian clock, and then try to put pieces together to figure out how those genes generate a clock. Now, this work, I have to tell you, at the time, this was 25 years ago, was a field that was not very well recognized. A lot of people didn't know much about it. And so it was sort of a risky project to be working on. The good news is that we actually were able to make advances. So I contributed to our understanding of how clocks are generated. And 25 years later, or almost 25 years later, it led to the Nobel Prize for my postdoctoral mentor, Michael Young, shown here. So he shared the prize with the heads of two other labs, Michael Rosbach and Jeff Hall, who had also made contributions to understanding how a clock is generated. And I'm sharing this story with you trainees because there's a lesson here. So I actually, so I was invited to Stockholm because I had contributed to this work. And I found, to my surprise and shock, that the poster put out by the Nobel Assembly showed, how the Earth is rotating. So therefore we have clocks because we are adapting to this daily cyclic environment we live in, and then how you generate a clock. And then my data were right here in the middle of this poster. And I have to tell you that when I generated these data, I did not believe them. Okay, when we all in science tend to doubt what we do, all right? Because you're doing it for the first time, right? Nobody else has done this before, so you doubt it. And in particular, if you're in a field where nothing or not much has been done before, there's no framework, so it's a real time of self-doubt. And I also felt like I had, in fact, it's true, I had very bad hands, so I never generated, pretty data. And so that made me question it all the more. And lo and behold, 25 years later, this is where my data are. And so this is a lesson to all of you who might go into science is, you never know when what you do today is going to be important tomorrow or 25 years from now. So this is the story of how you get clocks. Now, along the way, my lab has also become interested in one of the behaviors controlled by clocks, and that is in sleep. So I mentioned that sleep-wake is the best known rhythm controlled by clocks. But sleep, of course, is an entity worth studying in its own right. Like, why do we sleep? Why do we spend 1/3 of our lives in this state that is so non-productive? And we absolutely have to do it, because when you don't sleep, a lot of bad things happen. Okay, now this is sleep independent of its timing. Okay, yeah, sleep has a 24-hour rhythm, but that doesn't answer the question of, why do we sleep? And those kinds of questions come from sleep depriving people and asking, how are they compromised? And when people are sleep deprived, they show a lot of deficits in behavior, in learning and memory, in immune function, in metabolic function, in cardiac function. And so some years ago, we asked, we managed to use the fruit fly to figure out how clocks are generated. Can we use the fly now to ask why we sleep and what makes us sleepy? So you may have noticed that I kept referring to the fly rest activity cycle, because 20 years ago, we did not know if rest in flies was sleep. They rest, but are they actually sleeping? And so we put some effort into this, showed that fly rest is a sleep-like state. And we now are using the fly to understand how sleepiness occurs, why we sleep. And for both our circadian work and our sleep work, we are also now starting to translate this to mammalian models, including humans. And I will stop there. Thank you.
Well, thank you so much, Amita. And I hope that given the late hour for people in India, while Venkat is putting up his slides, I hope that's woken you up and excited you enough to keep you awake all through the night. So thank you very much. Sundar, I'll transfer this over to you then.
Thanks, Suresh, and for inviting me to this great symposium. And so I'm at the University of California, Davis on the West Coast. And the previous speaker, Professor Sehgal, gave an excellent excuse for me for this being very early in the morning. How now I understand why I feel the way I do now. So hopefully I'm still coherent enough that you can understand me despite my disrupted circadian rhythm and sleep. So we work on self-cloning plants by genome editing. That's the title of my talk. But our research really started with a very basic biology question. How does life begin for a plant? So life begins in plants in a sense the same as in animals. You have a sperm cell and you have an egg cell. The sperm is within the pollen, the egg is within the ovule, and they fuse. This process is called fertilization. And sperm plus egg gives you a cell called a zygote. Now this zygote is a special cell in that it now is able to undergo cell divisions and make an embryo within a seed. And furthermore, the seed then gives rise to the next generation of the plant. So everything that is here really began in these stages, and especially with this one cell that went on to make a whole organism. So it's a very special cell, and we got very interested in this and what makes it special. How we looked at, we did something called RNA sequencing, where we looked at the RNA produced by the cell. In the process, we found that there's a gene called baby boom 1, BBM1. This gene is active in the zygote. That's great. It's also strangely on in the sperm and off in the egg. And it's a regulatory gene. That means it's a type of gene called transcription factor, which controls other genes. Like these are often molecular switches in the genome. So here again, here's the egg cell. And these are sperm cells within the pollen. They're green because we've able to label the BBM1 protein, the baby boom protein, with a green fluorescent molecule. And now egg plus sperm gives zygote. And that also has BBM And of course, this zygote then goes on to divide and make an embryo in the seed. So we have speculated that BBM, which is on in the zygote and in the sperm cell, might be what the egg cell needs as a jump start to go through this whole program. Remember, the egg is what does most of the work. The sperm cells are tiny, and this much of the zygote is really derived from the egg cell. Okay, so then the question is, if that BBM1 is so important, that's what the sperm cell is bringing over, then if we could switch on BBM1 before it's fertilized, then can you bypass the sperm cell and just go on to make an embryo without fertilization? And the answer in short, in one word, is yes. And here's how it happens. So here's your egg cell. We introduced a transgene that by which BBM is expressed. It's in the egg cell, and we got an embryo. And this embryo was without fertilization. So it then went on to develop to make a plant, and this process is called parthenogenesis. So you got an embryo without fertilization by the sperm cell. The egg cell on its own was able to do this. You can also think of this as virgin birth in plants. Okay, so now switching on this one factor in the egg cell is sufficient to bypass fertilization. And this egg cell makes haploid progeny. That means haploid means one set of chromosomes. Here's an example of a haploid rice plant that we produced using this method. And it has only one set of chromosomes. Why is that? Because a process called meiosis ensures that every egg cell inherits half the chromosomes of the parent. This is true in plants, it's true in animals and in humans, that egg cells have half the chromosomes, and so do sperm cells, have only half the chromosomes. So then the question is, what would happen if we knocked out meiosis? Then the egg cell would inherit all of the parental chromosomes, right? And now if this egg cell could undergo parthenogenesis, you'd get clones. That is, you'd get progeny that are genetically identical to the parent. So how do you knock out meiosis? Well, fortunately, two collaborative labs, Mercier and Guerdoni in France, had found that mutation of 3 meiotic genes, which they called MyMe, eliminates meiosis. So that's what we did. We took this result and edited the rice genome for these 3 genes to block meiosis. If you do that, the result is diploid egg cells. So now instead of being half the number of chromosomes, these special egg cells have the full set of parental chromosomes. And here there's a schematic showing what we did. Here's the rice plant. We used genome editing to eliminate meiosis. That means you get a diploid egg cell. And now we then triggered parthenogenesis by using BBM1 to make an embryo, to make a zygote, which then made an embryo and then made a plant. And these plants, because they're derived from egg cells that have the entire set of parental chromosomes, these progeny are genetically identical to the parent. They're clones. And we've been able to do this for successive generations. So we took the clones, made clones out of those, and then further clones for 8 consecutive times, so that we now have the 8th generation, so the last sexual parent was 8 generations ago. And this is the 8th descendant clone. So this process is called synthetic apomixis, and it's published in this paper that you can look up if you want to know more details. Okay, so now I come to the application. Let's start with something called the hybrid revolution in agriculture. Here is a graph of maize or corn yields in the United States from the last century and a half. So from about half a century here, the yield, which is on the y-axis, was pretty much steady, flat. This is before hybrid maize. Once hybrid maize was introduced, see what happens. It goes up and up and up, 4 times, 5 times the yield that you had before. So all of this was done with hybrid corn. here's what's the situation with rice, though, is that most rice farmers don't grow hybrid rice. It's about 5 to 10% in India and 0%, close to zero in Africa. This is all the rice growing regions in the world. And yet, if you look at global rice production, we are going to need a lot more rice to feed a growing population, and to meet those needs, we'll need 20% more rice by the year 2035. So here's the amount of deficit that is being shown here. That's what we need to meet. And without using more land, this can be met by just growing hybrid rice. So why don't we grow hybrid rice now? And the reason is that hybrid seed production is labor intensive. That makes hybrid seeds for most crops, including rice, too costly for poor farmers. Why is it labor intensive? That's because to make hybrids, you have to cross the two parental lines that make the hybrid. And the hybrid is much more vigorous than either of the parents. Okay. Well, that's great, but now you take the seeds of the hybrids and plant them. What you find is that the progeny of the hybrids are not as good as the hybrid itself. So you get segregation, which means you get all sorts of sizes and shapes. And this is no good for high yields. You lose that hybrid vigor in the progeny. So so it's this process of crossing. So you can't just plant the seeds. You have to go back and recross these lines, which has to be done through labor-intensive methods. That's what makes hybrids costly. Now, instead of doing this, if you could just take the seeds from the hybrids and make them be as good as the hybrids, that would solve the problem. That's what we did through our method of, that is, we made clones, a method to make clones of the hybrids so that the progeny have the same genetic constitution as the parent, the same high yields. So this clonal hybrids produced by genome editing will allow farmers to produce their own hybrid seeds inexpensively, and they simply replant seeds that they've harvested. So that's very cheap compared to buying, say, seeds every year. And this has been subject of much discussion since the work was published. And here's from the something from the World Economic Forum, which you can read. It's an issue of January 2019, which describes the potential impact of this technology. And also, if you want a more scientific insight, science-based insight, there was an article in Nature Biotech, February 2019, that also discusses the same thing. And I thank you for your attention, and I'll end here.
Okay, terrific. Thank you so much, Sundar. Very exciting and a lot of potential. So I'd like now to engage in a panel discussion where I'm going to throw out a set of questions, but I'd like invite the panelists to also talk to each other and raise questions that you might have. And just to get the thing started, let me just ask each one of you, so Amita gave a wonderful presentation about how she picked a problem that she was not really sure that this was going to be significant or important. But then, curiosity and great experiments led to the fact that this ultimately resulted in a Nobel Prize for the people doing the work. And every one of us has similar examples. So how do you pick a problem? And in particular, what gives you the courage and the persistence to stay with a problem, working on it for 20, 25 years? Because sometimes, science can be frustrating. You go through periods where there's no results, and then suddenly there's something that's very exciting for the entire planet. So let's talk a little bit about that. I'm going to start with Amita, but please feel free to chip in and anyone can answer these questions. So Amita, just talk a little bit more about your work and when you got started with it, and when did you realize that this was going to be conserved in other systems other than flies, and when did you get really, when did it hit you that this is really a terribly important problem?
Yeah, so these things, as you mentioned, happen gradually, slowly in science. So at any given time, you may not know whether what you're working on is going to be relevant or not. I should say, that as a trainee starting out, there is a temptation to jump on the bandwagon of the latest, hottest thing out there. And I kind of caution trainees against doing that, because the importance of your contribution, will be perhaps less in that type of area where already a lot is known and it's, become mainstream, than one that is just developing. But it's hard. You take that chance and you wait for things to happen. In my case, I finished my postdoc, as in I went to look for a faculty position, and I actually looked for a faculty position before the bulk of my postdoc work was published, in part because I was tired of being a postdoc, in part, and again, this speaks to the self-doubt, I thought to myself, you know what, I should find a job before everybody finds out that this whole thing is not real. And, I swear that was like, going through my mind. And it wasn't until a year after my postdoc, we published the work and things suddenly in this field started picking up. And then 3 or 4 years later, we realized that it's all conserved in mammals. And a couple of years after that, human genes that were mutated and leading to circadian disorders in humans came along. So in this case, it panned out. There will be times when you start something and it doesn't quite pan out, and you have to use your judgment. I think the most important thing is that you be passionate about what you're doing. And as long as that is the case, going into something that's new, the potential for impact is going to be much greater.
Well, thank you. Let me turn to Vivek because he also worked in a lab in a field in protein secretion, which was very, very complex. It was not clear that one would get to a molecular understanding. And he was one of the pioneers in Jim Rothman's lab to crack that particular problem of how proteins are packaged into vesicles and transported from one compartment to the other inside the cell. So Vivek, tell us a little bit about the challenges you might have faced. Were you confident? I bet you were, that this would solve itself and this was an important problem? Your microphone is off.
I've always been very confident, but I wouldn't say that I fared well. You know very well that I had to try 2 times to get my position at UCSD in your department as a faculty. So when I finished my postdoc at Stanford, people said, yeah, so we will understand how proteins are secreted. And it has been a challenge. And the only advice I have for the young ones, since I'm not so young anymore, is that, work on something that hasn't been beaten to death, and don't follow fashions. There was a time when, microRNAs were very fashionable. Then, everyone was into stem cells. Then everyone got into the neurodegeneration. And now everyone is into these liquid condensates. What happens is when a topic becomes very fashionable, there's a lot of stuff that is done in a rush, and it is not necessarily of the highest quality or doesn't really move the field forward. It's just filling in the gaps. So the best is work on something that you really think about all the time. And how you do that, I don't know. It's just like someone who is interested in being a cricket player is not going to wake up one morning and say, oh, you know what, I'd like to have a go at tennis. I think it is coming from you. It has to come from you and your discussions with your colleagues and your neighbors, and looking up to a few people that you admire. I remember when I joined Jim Rothman's lab in 1985, 86, we had not a single protein identified in the secretory pathway. And Palade had basically laid out the pathway in 1974. And when Jim and Randy and Tom Sudoff got the Nobel Prize in 2013, I was also at Stockholm. And I remember these 3 legends were saying, everything is done. We know everything. And in fact, I completely disagree. I think we have nuts and bolts of the processes, but there are huge gaps. And this is always going to be true in science. So it's never complete. Thales said that 2000 years before Christ, that life science will never be complete, accurate, or permanent. So if you follow that motto and you believe that the currency of science is or our data, then you keep at it. Keep generating original data and hope that people catch on to it. It takes usually, a good paper takes about 5 years before its value is appreciated. And if you catch the attention by producing something unique that is different, then you will promote the field. And I think this has been my motto. And I still think that we're using the same thing. You know, 15, 20 years ago, in our field, we used to talk about, hey, how are collagens secreted? How are mucin secreted? Amita mentioned this business of melatonin being secreted in a circadian manner. In fact, collagens are secreted in a circadian manner. And some of the genes that we identified show circadian day-night expression patterns. Who would have thought? I would still like to know what happens when you sleep, you start secreting certain chemokines and cytokines. How does that system work? I was jotting down on a piece of paper when Amita was describing her findings. So I think this is just internal. It's like a clock. It keeps you going. And this has been my way of being. And that is, you take a problem that is not very well understood and you get into it. Surround yourself by very smart people. Think of yourself as the lowest on the ladder and look up rather than looking down, and keep at it.
I can assure you, in terms of the passion that our panelists show, the persistence about working on a problem does not undergo any kind of circadian rhythm. They're thinking about their problems 24/7. Sonia,
Yes.
you're younger in your career, getting launched off. What excites you the most? And tell us a little bit about this thought about, finding a problem that excites you, and how you go about following that course.
So, so I guess my take on this is that one needs to tread a fine line between persistence and so between persistence, knowing when you have to stay with it to finish the job you've started, and then knowing, listening to yourself and knowing when you've lost interest in something. So I think both of those things. So for me, if I wake up in the morning and I'm thinking about that problem, I know that that's a good problem to work on. I guess that's what I've done. And sometimes when you realize that, okay, I'm getting tired of this, you have to know when to stop, but also know when you need to complete that job and then move on. So both of those things.
Sundar, you have any comments to add? Particularly this transition with the clonal hybrids, very exciting science, but I guess this came more recently, and it's really revolutionary breakthrough. Tell us a little bit more about it.
Well, I don't have much to add to what the other panelists said in terms of, I think they all made excellent points, and that's the one thing I can add is that when we started this project, it was not an applied science problem. It was basic science. We just wanted to understand, how does an egg cell become a zygote? And there are other people trying to solve this problem more directly of clonal hybrids, just by more directed way, and for over 20 years, I would say and the solution came not through, oddly enough, not through directly approaching the problem, but through indirectly through insights gained from basic science. So even if so I guess my message would be, even if you're interested in applications or the impact of your work, don't neglect the basic science. That's what can ultimately get you there.
Yeah. So I'm going to, we are running low on time, but I do want to address one other thought, and this is aimed mostly at the listeners. So most of us were trained in very traditional sciences, meaning biology has always, for a long time, has been a descriptive science where you just describe phenomena. And it is only more recently that it's moving in the direction of a more quantitative, more predictive science. So in terms of training or teaching, can you talk a little bit about how each one of you has moved from your traditional field to learn other things, whether it's informatics or technological improvements in instrumentation? Let's talk a little bit about that aspect to see where is biology headed, and what would you recommend that a student doing biology today, should they just focus on botany or zoology or something like that, or do they need to have a broad interdisciplinary training to crack a particular problem? And anyone can start, really. Sundar, you can start. Perhaps, or Amita, go ahead. Yeah, please.
I just wanted to say very quickly that, yes, biology has become very quantitative. Much of it, I must admit, I'm not terribly happy about. I like to see things that are qualitatively different and not always quantify, but quantification is where it's at. Everything is digitized now. And my one piece of advice would be to students, take math and stick with it. One of the big mistakes I made during my training in India was I was allowed to leave math pretty early in my training, and I did it, and I regret it every single day.
Others?
Can I just add something too? So I was trained as a biochemist, both as an undergraduate and when I was at Oxford. And even at Stanford, I was basically a biochemist. And then we were the first ones to do one of the first genome-wide screens in Drosophila. And we are now dealing with patients with clinical features of asthma and mucinous cancers and tissue fibrosis. So I think one of the key features of the next generation of scientists has to be interdisciplinarity. I think you have to be fluent in many areas of biology or sciences. I think it's going to turn out that, we will have all the molecules that are functioning in a cell in the next 10 years or so. We will then be able to hopefully we'll get to the physics of the biological processes, which cannot always be simply explained in terms of molecules, but it will have to be in terms of forces, pressure, density, volume, viscosity. And Amita said that you have to be fluent in mathematics, which is very true. And unfortunately, I'm a complete zero, but I've surrounded myself with people who are very, very smart. So you need to understand bioinformatics so that you can now do gene building. You can build genes and ask, here is a protein, and I would like to find if this protein is present in bacteria as well. Well, it may not be a structural ortholog, but it might be a functional ortholog. And you need to be able to decipher that kind of information. You have to have a mindset that allows you to see beyond just the little area, whether it's molecular biology or it's cell biology or imaging. So don't be a one-trick pony. You cannot go very far with that because science is changing at an amazing speed. So this is my little penny's worth to the young ones.
Before I turn it over to the others to comment, I'll just say that all of us, including myself and the panelists, have surrounded ourselves with people more brilliant than we are, because that is the way to success. And in fact, we readily go and interact with people, talk about issues, and are happy to get ideas and input from other people. So this is, anyone who thinks they have a brilliant idea and doesn't want to share it with the rest of the world is likely to fail, because they don't get the benefit of input from the rest of the society and their colleagues. And you can certainly learn new fields and disciplines by having colleagues teach that to you. So Sundar or Sonia, do you want to quickly comment on this topic?
Sundar, please go ahead.
Oh no, sorry. I'm a bit sluggish here, but yeah, no, I think absolutely you need to have multiple strengths now to survive in biology today and to do well. And again, a grasp of computational tools is really important for anything that you do, at least in the general areas of genomics and gene interesting. And even beyond that, neurosciences, I think you can't avoid those things. You don't have to be an expert in math, but you just need to know how to use the tools of math. Yeah. And that to me is a critical thing. And I was a physics major. In fact, I started my PhD in physics before I switched. So, and what I found was that you couldn't directly model biological systems the way that I'd hoped you could, because biology is just too complicated. So it doesn't seem to follow any rules. So, but you can still do interesting work without, by a judicious use of computational tools. And you can't avoid that now. Yeah. You have to do it.
Sonia, did you wrap up this thing with your final thoughts on this?
I don't think I can add anything more. I'll just say that I agree with all the other panelists, and maybe just say that to the students out there, don't get put off or overwhelmed by this, computational math. If you're interested in a problem, I guess you'll follow it through, and you'll meet with, collaborate with all the right people who will be able to help you do the things that you want to do. So we all have our strengths, and we work with the people who complement our strengths.
Yeah, so Sonia, I think you put it very aptly. That is, you follow the problem, and if the problem is interesting, you will acquire all the tools from various disciplines to tackle that particular problem. And that's what keeps you going in this. So I know the time is short and it's late for the people in India. So I want to thank all of you for a very exciting session. It's a a la carte menu on modern biology, but I hope it has piqued your interest, and you can go look at their websites. And thank you again to the audience. And Shruti, I want to hand this back over to you and thank you for the opportunity to talk to this group.